Good morning. Can you hear me? I think. Good morning, everybody. Today we are opening the second session of MESMAP oral presentation. Our chairs are Violina Angelova from Bulgaria and Narin Sadıkoğlu.
In this session, Professor Ayşe Beklerci and Dr. Hayrettin Ozan is the
Professor Angelova, can you hear me? Please unmute your microphone.
Yes.
Now I will share the program. Our 1 minute. This is our program. We have six presentations. The first two of yours, and then Doğaçan from Japan, Narin Sadıkoğlu, Murat Pekmez, and Ibrahim Tümen. Each presentation 10 minutes. You can present your both presentation together. After all the presentation, we will have discussions. You can introduce yourself and start your presentation. Thank you.
Okay. Okay. I tried to share my screen, but maybe it's a problem.
First, open your presentation on your screen, and then share screen bottom of the screen.
Okay, I opened the presentation. It is on my screen, and after that,
share screen bottom green. Share screen bottom of the screen. Zoom, come to Zoom program again. Okay. After you're opening your presentation, come to Zoom program and share screen, green one.
Just a moment.
Now,
okay, enter in the Zoom because it's not possible.
Yeah. Now try again.
Again, enter in the Zoom meeting.
Yes.
Okay.
Share screen, green, bottom of the screen. Share screen.
Share. And here, it is one of the presentation. Can you see?
No, you should choose your presentation. If only open your presentation on your computer.
Yes, I open. It is on my computer, but I cannot share computer sound. Share, okay. Can you see
coming.
now?
Now coming.
Yes.
Full screen, please.
A full screen?
F5. You can
a slideshow. Do slideshow, okay.
F5.
Okay, can you see? It is the second presentation because I opened this the second presentation.
No problem.
Okay.
No
this presentation. Thank you. First, I want to thank you for possibilities to take part in this symposium, and I present some of my results. The first are the results from basil, heavy metal accumulation and composition of essential oil of basil, cultivated on contaminated soil. My name is Violina Angelova, and I'm from Department of Chemistry and Phytopharmacy from Bulgaria. I tell you some words about the phytoremediation. It is technology. With this technology, we can clean the soil. And this technology has a lot of benefits because we can do this technology in situ, and we. Sorry. Okay, and it is low-cost technology. So the problem it is to find suitable crops for this phytoremediation. The food crops it is not good choice because heavy metal can enter in the food chain upon consumption of this culture. That's why the medical and aromatic plants can be a better choice for phytoremediation because these species are grown primarily from for processing. We obtain essential oils and contamination in food chain with heavy metals is eliminated. The problem in Bulgaria, here it is the map of Bulgaria, and we have a lot of spaces which are contaminated by heavy metals. You can see the area which are contaminated. It is 0.7% of agriculture areas in our country. This area are contaminated 5 times above the concentration, and the pollutant are lead, cadmium, copper, zinc, and arsenic. And here are the places: Kırjali, Plovdiv. Plovdiv, it is my region. Sofia, Pernik, and we have in Madan, Rudozem, Zlatograd, different places which are contaminated by heavy metals. We have 2 problems. This is the non-ferrous metal works. In this place, close to this place, I make my investigation. We have 2 possibilities for contamination of the plants, and here you can see the first way it is through the uptake of soil accumulated heavy metals by roots, and next it is absorption of aerosols fallen of leaf surfaces. This concentration of aerosol change during depending of the source distance, the weather conditions, and of course the size of particles. When we speak about the heavy metal uptakes per plants, there are 2 factors. One is soil factor. The heavy metal accumulation by plants depends of heavy metal content in the soil. Some parameters, soil parameter like capacity, redox condition, organic matter, and so on. And the second factor, it is plant. So we must choose the proper plant when we want to do this phytoremediation. The aim of this work it is to determine the content of heavy metals and of course chemical composition of the oil, and as well to identify the possibilities of basil growth on soil contaminated by heavy metals. Basil, it is an herb in the mint family. It adds flavor to meals. Usually, this plant play a role in our cuisines. It is possible to use basil to prepare pesto, and it is to add like flavor to salads, pizza, and other dishes. We have 2 forms of basils, green color and red color. In this presentation, will speak about the sweet basil with green color. The weather in our country, it is very suitable to grow this plant in at home and in the to use in the garden. We make field experiments and the plots were situated at different places, distances from the source of pollution. And we use block methods with 4 replication, and the size of the test parcel was 100 meter quadrat. And the plants were grown in accordance with the standard technology. We use 50 plants for analysis. We make soil analysis, total content of heavy metals, and DDPA, this available form of heavy metals. And of course, plant analysis. The plants were gathered when they reached the flowering stage, and the content of heavy metals in different parts of the plants was determined by ICP. It is inductively coupled plasma emission. From the above ground of the plant in the laboratory conditions, we obtain oil by steam distillation, and after that we make analysis by gas chromatography, the mass spectrometry technique. Here on this screen, you can see the result of heavy metals in the soil. The soil it is contaminated. Here on the last row, you can see maximum permissible concentration from Bulgaria. The total content of lead is almost 1,000 milligram per kilogram, and the maximum permissible concentration is 80. You can see that the soil is very polluted, and it is the same about, if I speak about zinc and cadmium. The total concentration is approximately 2000, and the cadmium it is almost 30. The MPC value it is 2.5. So the soil it is very contaminated. Here it is the explanation. In the table, you can see except total content and this DTPA content, this value explain us how much heavy metals from soil can be moved to the plant. So it is very important to know the available form of heavy metals. The next table, it is the results from heavy metals in basil. We make analysis of fruits, stems, leaves, inflorescence, and oil. And you can see that most of the heavy metals accumulate in the root system, and the less are accumulate in the leaves and stems and inflorescence. The oil is not contaminated. It is good because we can use the oil for different purposes. Here are the explanation about this table. So I move. The content of heavy metals in the oil were lower compared to the above ground part of the basil, and the quantities of the heavy metal in oil are lower than accepted maximum permissible values, and they meet the requirements for ecology clean product. This means that we can grow basil plants on the contaminated soil, but the product it is clean and we can use for different purposes. Here on the next slide, you can see distribution of the heavy metals in the basil, and I mentioned that the most of the heavy metals are accumulated in the root system. The basil oil, we analyze basil oil, and we found 30 components in the oil. And among the detected compounds, beta-linalool, it is in the major amount. You can see here the other component. And our conclusion are that the contamination of soil did not affect the quantity of the oil and quality of different amount. So the basil can be a valuable product for farmers from these regions. When we make classification of the compound, we can see that the highest content is the content of oxygen-containing monoterpenes, followed by sexy terpene hydrocarbons, oxygen-containing sexy terpenes, and monoterpenic hydrocarbon. But the bigger fraction It is oxygen containing monoterpene. It is a red color. And conclusion, basil, it is a plant which tolerant to heavy metals and can be grown on contaminated soils. Heavy metals do not affect the development of the basil and quality of the oil. The possibility of industrial processing will make basil economically interesting crops for farmers of phytoremediation technology. It is my first presentation.
Thank you. Thank you, Professor Violina. And you can attend the second one, and then we can make discussion.
Okay, I will start to again to
Yes,
share.
I stop. Yes, you share again, please.
Okay.
Another one.
First to open.
Open first.
Yes, I just want to. Okay. Here it is the presentation. Maybe it's necessary again to start the Zoom meeting.
Yeah, Zoom again.
Yes.
Share screen and full screen.
Okay.
Yes.
Can you see? Okay.
Full screen.
Yes, I forgot about the full screen.
Great. Thank you.
Yes. Okay. Just a moment. 1 second. Okay. It is the second presentation. It is about accumulation and composition of essential oil of juniperus. But this time, these plants were grown naturally on serpentine soils in Bulgaria. Here we have different pollution. It is mostly by nickel. And here I first start with explanation about these soils, because most people doesn't know about this soil. These soils are naturally soils, and they derived from ferrous or manganese-rich ultrafamic rocks. And they are enriched with some metals like nickel, chromium, and cobalt. They have abnormally high concentration of these elements. And some of the toxic metal are in the minor amounts. So the plants which are growing on these serpentines have a remarkable ability to absorb and accumulate inorganic compounds, components from this media. The aim of this work, it is to terminate the heavy metal and micro and macro elements and chemical composition of the leaves and berries of essential oil of juniperus growing on this serpentine soil in Bulgaria. There are serpentine soils in Greece, in Albania, and Turkey also, not only here in Bulgaria. They are very interesting. These plants, I'll explain some words about these plants. It is small tree or shrub. It is native to the Mediterranean. And these plants need warm Mediterranean climate. And it is possible to found in dry or rocky or sandy soils. This tree is able to tolerate colder climate and grows in elevation from sea level up to 2,200 meters. We can obtain oil from berries and needles. And this oil is natural product, and it's possible to use in different industry like food industry, perfumery, cosmetics. Usually we use hydrodistillation when we obtain this oil. The health of oil depends of the plant's geographical location, degree of ripeness, age, and different condition. Here, it is explanation which are the places in Bulgaria. I use some serpentine sites in eastern and central Rhodopis. It is the bottom of the map. We choose 8 serpentine sites. They are located close to Kazakh. It is very close to the Greece border. Kaminyane, Chichevo, and so on, 8 places. For analysis, we take 13 soil samples from these soils, and we make analysis of the soil. We use one of the method. It is acid digestion with aqua regia. It is with hydrochloric and nitric acid. Again, we use ICP method for determination of the total elemental concentration in the plant and, of course, in the soil samples. The plant samples are collected from these serpentine soils, and the content of heavy metals in leaves berries was determined by ICP method. Those were obtained by hydrodistillation and analyzed by gas chromatography, mass spectroscopy technique. What is interesting for these serpentine soils, they are shallow soil, and here we see deficiency in nutrients. In these soils, they have a high magnesium-calcium ratio. In agriculture soils, this ratio, it is calcium-magnesium. So calcium, it is in the bigger amount than magnesium, but in this serpentine soil, we can see opposite. And we have, of course, high concentration of toxic metals like nickel, chromium, and cobalt. In this table, you can see the total concentration. It is minimum content, maximum content, and average content. The salts are contaminated by nickel. It is approximately 2,400 milligram per kilogram maximal content. In Europe, we can see and found the nickel contaminated soil, and the content will be around 3,000. But in Bulgaria, the soils are with less amount of nickel. And you can see here that magnesium content is bigger than calcium content. So the ratio magnesium-calcium is over 1 for magnesium. Here in this table, you can see the content of the metals, micro and macro elements in the junipers. Despite that these plants are cultivated, they are naturally grown on this contaminated soil. The nickel content is very low. It is in the leaves content, and here we can see in the berries content. And you can see the amount of calcium with magnesium. Here in the plants, the content of calcium is bigger than magnesium. It is very interesting fact. And here I'll explain about why this is the fact. Because the plants have very efficient calcium uptake system, or ability to exclude magnesium from soil despite high concentration in the soil. The concentration of nickel, it is high, and it is elevated from the samples from the serpentine soil, but not so high. About the other element, when we speak about the plants, there are rule. The nickel and chromium content must be between 0.4 and 4 milligram per kilogram. It is for nickel, and for chromium between 0.2 and 1 milligram per kilogram. Our results about nickel, it is a little bit higher. For example, for berries, it is 2 times more. The content in chromium is lower than these values. The trace elements are in a small amount, and here you can see the manganese concentration in the juniperus were below the critical level. Then it is the values between 3,000 and 300 and 500 milligram per kilogram. About heavy metals, there are recommendation about heavy metals. So if we grow plants on contaminated soil, they must have low concentration of the element. And here are the data about arsenic, cadmium, and chromium. So in lead, in our cases, the lead concentration were below 1 milligram per kilogram in berries. And of course, about cadmium, again, we have the concentration less than proposed in the European Pharmacopoeia monograph data. So these plants can be considered as excluder plants because these plants contain a low concentration in the parts, aerial parts, even in case of high elemental concentration. And metal concentration for toxic element in plants and oil were below these limits for purposes. That's why we can we make recommendation about junipers that we can collect the above ground parts of the plants and to produce oil and to use for different purposes. So this elevated nickel content does not affect the development of juniperus and quality and quantity of the oil. The health of the oil, it is 0.1 and 2.2 if we extract from leaves, and the second, it is when we extract from berries. We found different compounds. When we extract oil from leaves, so we found 60 compounds, and we have used berries. We found 39 compounds. Here are the composition of the oil from leaves. The limonene, it is the major component, and of course we have the other. You can see the percentage of different compounds. It is about leaf oil. For berry oil, we found that myrcene and germacran D, it is again alpha-pinene, limonene, and the other are the major component in this oil. Thank you very much.
Thank you, Professor Violina. Is there any question to Professor Violina? I think there is no question. Thank you. I want to give you the chair, and I said you can present the presentation. 1 minute. Yes, here. OP 15.
Yes.
Yes, you can present, and now you are chair. You and Narin Sadıkoğlu, you are two chairs. I give you chair. Okay.
Okay. The next presentation, it is phytochemical analysis and habitat suitability mapping of, I cannot see, it's
Glycyrrhiza glabra.
Yes, collected in one Hatay region of Turkey. And the present author, it is Al-Sadi.
From Japan. Yeah.
From Japan.
Yes.
Okay.
Thank you very much. Once again, hello.
Hello.
My second presentation is about the Glycyrrhiza glabra that collected from Turkey and their habitat suitability. So as we know, Glycyrrhiza has a wide application in food industry, in cosmetics, healthcare, like traditional medicine, hair care, due to its pharmacological properties and also sweet taste. There are two species that has a high value in Japan, that Glycyrrhiza glabra and Glycyrrhiza olanensis. And these species undergo quality control before marketing. And this is there's a standards in the pharmacopoeia, Japanese pharmacopoeia, Korean pharmacopoeia, and also Chinese pharmacopoeia for the quality control of these species. So the marketed species should have at least 2% of glyceric acid before marketing. And the Japanese name for this species usually called kanzo, and the Turkish name, if I'm not wrong, is mian. So glyceriza has a lot of active component, very useful. That is glyceric acid, glabridine, leucotriene, isoleucotriene, and so on. And this has a lot of pharmacological properties. And in Japan, there's many products, marketed product that has isolated compound from glyceriza. These are some examples of these products. We have a health growth trigger serum and moisture whitening cream, hand cream, and combo medicine, and also some tea and a weight losing tablet. And because it has a wide distribution around the world, it has also variation in the secondary metabolite, and may be affected by the environmental factor. So sometimes it has some challenges to cultivate a high quality glyceriza that has a high percentage of glyceric acid and so on. So we try to utilize technologies like drone imaging, GIS, and Internet of Things, and especially for glyceriza, that natural resources of glyceriza, like Turkey. So here is the Turkish map, and this is from Tobavis website. It has a blend database of Turkey, and as you can see, there is the Glyceriza glabra has two variants and distribution all around Turkey, and in Hatay, it also distributed. So we want to quantify the glyceric acid content, glabridine, and leucotriene to check the quality of the Turkish glyceriza. And also we want to develop a geospatial model for the habitat suitability based on the environmental factor, and define also the factor that affecting the bioactive content of the plant. So we have a field investigation and data on the data analysis, and also this data will be used for future cultivation. So again, we have an in situ soil analysis using soil pH, soil bearing capacity, volumetric soil moisture content, and also we utilize the weather information from the Turkish Ministry of Agriculture and Forestry. And as you can see, also the study area here from these maps in Hatay region of Turkey. So we use the HPLC to quantify the chemical content of glyceriza, and we also compare it to the Japanese sample. Some of the sample cultivated in Japan, Glycyrrhiza glabra, but unfortunately the activity is very low compared to the Turkish one. And there is a marketed Japanese sample actually imported from China and has a high glyceric acid content, but also compared to the Turkish sample. And here is the HPLC analysis. And from our analysis, we found that the liquidine content was between 0.2 to 2% around, and the glyceric acid content between 0.5 to 2.4, and the glabridine content between 0.02 to 0.3%. For glyceric acid, some sample met the Japanese pharmacopoeia standards, that it has more than 2% of the glyceric acid, but some sample didn't met that. And the glabridine content was also very low, but this is not only for Turkish sample. Japanese sample also has very low glabridine content. So we check ANOVA and MANOVA statistical analysis to see the affected parameter on this content. And we found out that glyceric acid and liquidine content was affected by soil parameter, like a soil bearing capacity and the volumetric soil moisture. And the geographical factor, also the slope, aspect, curvature, and elevation has some effect on the glyceric acid and liquidine content. However, the glabridine content wasn't affected by any parameter. And according to some study suggesting that the glabridine actually affected more by genetic reason for the high or low level of glabridine content. Here the thematic map for the study area, considering the soil parameter, the environmental and geographic parameter. And this is the final habitat suitability zone map for the using a frequency ratio model. And this based on the occurrence and non-occurrence of the plant, and also for the multiple variable we have. And here we utilize, of course, the geographic, meteorological, and soil data for the GIS study. So we find out that some bioactive compound in glycyrrhiza was affected actually by the environmental factor, but some weren't. And we identify some factor that affected the bioactive content. And maybe in the future, this can help to obtain this high quality product. And that means has met the Japanese standard for 2% or high. And also we determine the suitability of the land for the plant cultivation. Based on these data, maybe we can use like a very high suitability zone for the future cultivation of the glycyrrhiza, and also utilizing the IoT device and sensor for this monitoring. Thank you very much for listening.
Violina, Professor Violina.
Does anybody have any question?
Yes. Yes. Do you have some question?
Yes,
I have.
Please. Yes, please.
Okay.
Thank you for kind presentation, Doğa-san.
Thank you very much.
It was great result, and we are
Thank you
together
very
in
much.
this study. I'm worried about
Yes.
if licorice is useful for COVID or not.
Liquidine.
Licorice
Ah,
products is useful for COVID treatment or protecting COVID. Is useful or not? What do you think?
we didn't try yet, but many people now trying the traditional medicine to check for the COVID. So maybe it has a potential, but we didn't try yet. We didn't check yet for that.
So we have review on the COVID protection and treatment, and you know about 2,500 Chinese traditional medicine. Also, Japanese Kampo medicine includes licorice in their recipe. So it's important to protect your throat.
Yes.
So we know that licorice also protect your throat and kill the viruses and microbes. I say that I believe that licorice is very, very important to protect yourself from the COVID
Yeah, maybe also like some isolated compound like glyceric acid or glabridine or liquatine maybe are the ones that are responsible for that. It's also interesting because glyceric acid using combo for the stomach problem and sometimes respiratory. So it's interesting. Maybe it has something.
Maybe we should
yes, we research.
We should work.
Yes.
Okay.
Yes, thank you.
Can I explain something too?
Surely. Takashi-san,
Yes,
welcome.
so now we are looking for another stage using COVID-19. Because of the Turkish plant materials going for the first stage, we like establish something to using the bioactivities. Then now, maybe Najim-san knows about ACE inhibitors. Relation with the COVID-19, the more effective.
Yes.
Yeah.
So therefore, Dosa and my team and using the Najim-san's material for the just try to the checking about the more effective with the combination with like Gushiriza for the future, near future, now coming up.
Near future.
So please wait.
Near future. Very soon. Very soon.
So
Inshallah.
we already know what to do. So
Inshallah.
we must
Inshallah.
stop the COVID-19 anyway. Thank you.
Okay, thank you,
Okay,
Takashi-san.
thank you.
Dr. Viola?
Yes, the next presentation. Thank you very much for your presentation.
Thank you very
it was
much.
very interesting.
Thank you very much.
The next presentation, it is new D2 Homes identification key for Salon-A genera, and the presented author it is Narine
Sadıkoğlu.
Yes, I'm sorry about the
I
I present myself. Thank you. Okay. I'm from Turkey, Department of Pharmacognosy, İnönü University, eastern part of Turkey. I present you a new dichotomous family key of Laeviaceae family. New name is Lamiaceae. It's a very big family, very large family around the world. Approximately 9 over the now 9,000 taxa now. It is the gene center of Turkey, is the gene center of the Lamiaceae, and 10% Lamiaceae members are live in Turkey around the world. In Turkey, we have flora of Turkey, and when the Lamiaceae family occurs, is published at in 1982, and this family represents by 45 genera and 713 taxa. Then many new taxa added, and a new supplement is needed, and it's published in 1988, and it contains 17 Lamiaceae taxa. Then continuing new taxa and new combinations, and the second supplement is published in 2000 and contains 27 taxa. Since the publication of Flora of Turkey, many taxa are still added to Turkish flora, and status of some species have been made synonyms. In recent years, due to increasing molecular and systematical research, amount of taxa and taxonomic categories are changed. Then a book was published. It's a list book, and it comprises 46 genera with 749 taxa in Turkey and very high endemism ratio. Many new taxa have been published and some taxonomic and nomenclature changes have been done, and many publishing after that, combining diversity of Turkey, many publication about this subject, and according to them, it is represented by 48 genera about. But after that year published, the data is not true because many changing have become. So, according to most recent data, there are 45 genera again. In recent years, the flora of Turkey, while adding many new taxa as a result of rapidly increasing systematical and molecular research, the status of some genera, some taxa, are being changed and become synonyms after publishing the second supplement. The current Lamiaceae key is not sufficient to identify the genera. For this reason, any revised identification key is necessary in Turkey. The key was first produced from that time in flora of Turkey and the East Asian islands. Identification keys are prepared as dichotomous. But multi-access keys are most useful than dichotomous ones. Therefore, firstly, a multi-access key was prepared and adapted to recent times. And then it was presented in Advanced in Lamiaceae Science Symposium. And however, a dichotomous key was prepared for identifying easily, but it has become insufficient due to recent developments. In this study, it has since then rearranged with the additional taxa and adopt the APG system. It was tested variously herbaria in Turkey. The genera names are sorted by alphabetical order. This is the list of Turkish Lamiaceae genera. It's 45 genera. This is the new key. And I have limited time, so I quickly tell. And this one is for beginners version because they have both synonym names as you've seen there, and then will be deleted when a person is professionalized. I quickly and in Lamialis, the old delimitations of Acanthaceae, Lamiaceae, Scrofulariaceae, etc. Were contradicted by molecular studies, and although still using many of these names, their descriptions are now very different. And several genera transferred from Verbenaceae to Lamiaceae in 1990s based on phylogenetical analysis and morphological and molecular data. The genocial morphology, palynology, and phylogenetic analysis of non-DNA data may be similar, so a considerable number of verbenaceae is now included in LAME-assay. Those two families, previously considered close but separate, can now be more easily identified morphologically than before. For example, Clerodendrum and Vitex were previously placed in Verbenaceae. Now they are considered in Lamiaceae. However, they have drupe type fruit. Unlike the nutlets in Lamiaceae, also flowers are similar but not the same. On the other hand, Vitex and Clerodendrum genera are transferred from Verbenaceae to Lamiaceae in 2009. Clerodendrum has morphological variation between species, so has included in different genera, even families, in the past years. Molecular systematic research conducted today still changed the status of the Clerodendrum genus. In 1982, Lamiaceae has 45 genus and flora of Turkey. Studies of molecular systematic caused Widemania galobdolon to be included in Lamium, Calamintine acinos in Clinopodium, and Corydothymus in Timbra, and finally Doriostoecas and Rosmarinus in Salvia. So coincidentally, again, 45 genera. Covers moved species of Leonorus marobiastrum to Chyturus marobiastrum in 1999. This name is Bezionum. Chyturus was added to Flora of Turkey in 2012. Phlomoides near former Phlomis tuberosa, many former species of the genera Phlomis and Aromastachys, of which 3 species are moved to genera for Flora of Turkey. The Bezionim names under the names. The genus Betonica is re-established and confirmed as the same. And also Pseudodignanus as the same. Perilla is a genus which is possibly typic and mainly found in East Asia. Perilla frutescens is widely distributed and cultivated in Far East, but is also found in Northwest Iran and the Caucasus. Though much found in Artvin, Turkey, a new species in 2002. This was the first time this genus was recorded in Turkey. As a result, a new genus was added for the flora of Turkey. Then Dirmenge found and described a new species, Lophanthus tursicus, in 2010. This was the first time the genus recorded in Turkey. And the genus Lophanthus has 23 species and most distributed in China, Mongolia, Afghanistan, and Central Asia. Recently, Sarpushan did a molecular phylogenetic study on Hymenocrater, Nepeta, Lophanthus, and other closely related genera and made some taxonomic changes among studied genera. For example, they transferred Lophanthus tursicus into Nepeta. According to Sarpushan, the complete DNA consequences of Lophanthus species with NEPED and hemocrater may provide more resolved phylogenetic tree and more accurate interpretation on taxonomy of the genera. Until having more data about Lophanthus tursicus, it is still accepted as a valid species in this study. Also, 2 new endemic species have been published from Turkey, not genera, but it's little important. And finally, in conclusion, taxonomic position of Doristotes hastata, Rosmarinus officinalis, and Lophanthus tursicus are still under discussions. Their final taxonomic positions will be clarified with additional molecular and systematical studies. At the present time, LEMES has 45 genera in Turkey. In this study, as an alternative to the key in flora of Turkey, which has lost its functionality since 1972, when it was prepared, a more useful diagnostic key was prepared for the Lamiaceae family today. Thank you. And is there any question?
Yes. No, there is no question. Thank you very much for your presentation. The next presentation, it is potential application of plant lectins against COVID. And Murat Pekmez will present. Are you here? No, maybe it's not here.
Murat
Okay.
Bey.
Murat?
No.
No, maybe it's...
Yeah.
Okay. And the last presentation, it is in investigation on effects of ontogenic variations of maritime pine on volatile components. And the presenter will be Tumen.
Ibrahim Tumen.
Yes. Okay.
Thank you, Violina.
Mm-hmm.
Again, greetings, my best greetings to everybody. Now share screen. Excuse me. Yeah. How can I? Now is okay?
No.
Hocam F5 yap.
Slideshow.
Okay.
Bir de yukarı çıkart sunumu. Şu anda paylaşılmıyor.
Nasıl
Ben
yukarı?
bir iptal ediyorum, tekrar bir şey yapacağım.
Okay.
Now is okay?
Yes.
Okay, thank you. Our presentation about maritime pine volatile components with my colleague Mr. Kurtça, Dr. Kurtça. Okay. As you know, maritime pine is a pine species which doesn't grow in Turkey, so imported and grown in our country in Turkey. Very big area in this country. We also know is exotic tree species. Botanical features, it has 20 and 25 meter heights and paramedical structure, and also needles 10-20 centimeter, okay, and cones approximately 9 centimeter, okay. we can see this maritime pine. The distribution area in maritime pine. Also in Italy, in another site. This pine species naturally grows in South Atlantic coast of France, Lens area, north of Lisbon, Corsica, and northwest coast of Italy, Morocco, Algeria, and Tunisian. Also in Turkey, in Black Sea, Marmara, and Aegean region. Also this. Also, we have a limited time. I have to faster. Yes, sorry. Also, these main uses for sound timber of maritime pine, joinery, both interior molding, skirting boards, flooring, and paneling. Also, also the maritime pine include the resin, is popular resin. Resin is the most important non-wood product from Pinus pinaster, direct or indirect after distillation. Turpentine and pitch oils, varnishes, adhesive, wax, and soap. The bark of Pinus pinaster may be distilled to produce tar or chipped and composted to produce low white substrate for nursery containers. Also, Pinus pinaster stands also are in ideal ecosystem for the development of edible fungi. Also, such as Boletus edulis, Boletus pinicola, Lactarius delicius, and Lactarius semisancophilus. Also, there are a lot of working pharmacological uses to turpentine obtained from the resin of all pine trees, antiseptic, diuretic, rubefacient, and vermifuge. Okay. Also, you see pycnogenol produced from maritime pine. Also, pycnogenol is very popular in the shopping center. Nutrition, also pycnogenol using as a nutrition supplement. Okay, it has been reported pycnogenol strengthens the immune system, helps reduce aging effects, and strengthens connective tissue and prevents skin aging. Also, we know COVID periods, pycnogenol, they can use pycnogenol. In our work, consent needles belonging to maritime pine that were grown in Barton region were used to experimental materials. We were collected from the same trees in 4 different seasons because we studied and searched. Also, we investigated seasonal ontogenetic variations. Also, the trees from which samples are taken, maritime pine. Also, hydrodistillation. Also, as we know, the essential oils example were obtained by hydrodistillation, HD distillation. We used Klevenger apparatus. Okay. Approximately 1 kilogram partly crushed fresh cones and needles we used. You know, Klevenger apparatus. This European Klevenger. GCMS analysis, we used GCMS Shimatsu, GCMS quadrupole instrument equipment. Also, the GCMS conditions we can see. Resultant discussion. Essential oil yields, needle and cones collected in different seasons. Also, this seasons November, February, May, and August. On the needle, we can say in August the high oil yields. Also cones in high oil yields. Also, we can say this yields in this summer and also the hot days. Also we can avoid these yields. Also in winter, approximately needle and on the needle, February and August approximately similar. But cones not similar, very different yields. Essential yields of needles and cones on the graphical. Volatile components, needle and cones collect different seasons. Main components, alpha pinene, see, needle November, cones May. Yeah, we can see on the months. Beta pinene, beta phellandrene, alpha terpineol, main components. Junipen, also trans beta caryophyllene and germacrene. We found 102 compounds, volatile compounds on the maritime pine. The main volatile components amounts of needle we can see on the graphical. Alpha pinene, trans beta caryophyllene, germacrene de main components on the needle. Also, another graphic, main components alpha pinene, beta pinene, junipen on the cones. As you see, in the May, alpha pinene is high. Monoterpenes, sesquiterpenes, diterpenes, and the others, 4 seasons, needle and cones, we can see. Of course, we know the plants mainly include monoterpenes. Also, we can see. In needle essential oils, main components have been detected as alpha pinene in November, February, germacrene D, trans beta caryophyllene. Also in cones essential oils, main components have been determined alpha pinene, beta pinene, junipen. In literature, it is seen that needle volatile oil were most examined in the volatile oil. Humachi et al. Components are beta caryophyllene and alpha pinene. Ismail et al. 2013 main components alpha pinene, beta caryophyllene. Also Ottavioli et al. Yeah, we see. Also in cons volatile oil. The seasonal change of volatile components of the maritime pine was rarely found in the literature. Yeah. When our studies compared to this study, it is seen the results of periodical change of volatile components in our study are similar to the results of Araval et al. 2014. Thank you very much. Now, are there a question or? Thank you, Violina.
Thank you very much for your presentation. Do you have any question? Can you hear me? Do you have any question?
I think there is no question.
Okay, thank you again.
Murat Pekmez is here. Nailin
Yes,
hocam.
I am here. No. I will share the screen, I think. Yeah.
Nailin hocam, Murat Bey'i anons edebilir.
I presented. The next speaker is Murat Pekmez, and his speech is potential applications of plant lectins against COVID.
Okay, thank you very much. I am from Istanbul University and Department of Molecular Biology.
Hocam full screen.
I would like to present my talk, potential applications of plant lectins against SARS-CoV-2. I would like to first give some basic informations about this disease and the virus, and then the usage of the lectins I would like to mention. So all of the people knows that this coronavirus disease, also it is called COVID-19, is a type of viral pneumonia caused by the severe acute respiratory syndrome coronavirus 2. It is also called SARS-CoV-2. And as you see in the slide, all of the viruses are the human viruses, but we know that this COVID belongs to coronavirus family. It is a large family. The most important characteristic is single-stranded RNA viruses. They can both infect animals and also humans. But in the slide, you all see the human viruses. The red ones are most dangerous ones. You know, also you heard about the MERS-CoV and SARS-CoV. Also nowadays SARS-CoV-2. These are the most dangerous ones. The gray ones on the left side, these are not mostly dangerous, but also affects humans, we can say. When we look at the classification of the coronavirus, the most large family is Coronaviridae, the subfamily is Orthocoronaviridae, and the genera it belongs, and SARS-CoV-2 belongs to beta coronavirus genera. And about the classifications of the, or identifications of the most dangerous viruses, coronaviruses, we can say, let's talk about first SARS-CoV. It is first identified in Guangdong. MERS-CoV is identified in Saudi Arabia, and COVID is identified in Wuhan, China. The spread in the countries, it is most interesting. COVID-19 is spread in a lot of countries. You see 210 countries. And you see the rate of the deaths. MERS and SARS cause much higher death rates, but total deaths is higher for COVID-19. And the most important problem is about the curing. There is no available cure up to now. And as everybody knows, self-isolation is the best preventive measure of cure, we can say. These are the values or numbers that I take yesterday. So at the moment, total deaths is approximately 1 million all over the world. And you see the total cases and you see the recovered people. When we look at the cases distribution, the United States is the most leading country, we can say. The second is India, and then Brazil, and so other countries. So what is the viral structure? It is also important for the therapeutic applications, maybe. There are 4 major parts of the virus: the membrane M protein, spike protein, the nucleocapsid N protein, and envelope protein. Also, the genome is RNA genome, and the most important enzymes, we can say, hemagglutinin esterase. This is also very important for the spread of the virus to another host, maybe we can say. So I will talk about detailing the spike protein. From the slide also, you can see the branch parts of the protein. This all branch parts refers to N-glycosylation, N-link glycosylation parts. And also NTD is the N-terminal domain. This is the RBD, the receptor binding domain, and fusion peptide, and so on. You see there are nearly 22 N-link glycosylation parts, you see. And the protein is formed from 2 parts, S1 and S2, 2 different units, we can say. So this is the also, I will not go into detail, but this is the mechanism of the viral entry and replication of the virus. And after the formation of the mature variant, by exocytosis, the virus can spread to other parts or other hosts, we can say. So let's come to the major therapeutic strategies. There are a lot of therapeutic strategies, as you see. Nowadays, there are a lot of researchers trying to find some vaccines, but also the other strategies are available. For example, modulation of the immune defense supplements and nutraceutical supplements, oligonucleotide and gene therapy, blocking polyprotein post-translational processing, interfering with the endostatic pathway, blocking virus cell entry. Also, this is very important. I will mention later about talking about the lectins, and targeting cellular signaling pathways, and also targeting viral RNA-dependent RNA polymerase. So as you see, there are a lot of therapeutic strategies, but today we will shortly focus on the plant lectins. So what are plant lectins? The plant lectins are proteins. They are carbohydrate binding proteins, and they are capable of recognize some specific parts on the proteins, and the binding is reversible. So this is also important. You see an example of plant lectins, Viscum album lectin. So let's see classification of the plant lectins. The most important part is carbohydrate specificity. It can differ from lectin to lectin. The major structure, the structure of the lectin is also important, and the family. This is the example of a legume family, and this is a short chronological or historical, maybe we can say, the start of lectinology. It first starts by isolation or finding the isolation of hemagglutinin protein, a specific lectin, ricin, it is called. You see, it is very old, 1888, and a lot of different steps was produced by the researchers, and it comes to the today's lectinology or glycobiology, we can say. So how can we use the plant lectins against SARS-CoV-2? The most important characteristic is the antiviral capacity. Nearly all of the plant lectins has some different antiviral activities or capacities, and this affects by binding to the viral envelope glycans, and because of this, it prevents the entry of the virus into the cell. For SARS-CoV-2, we know that these spike proteins are responsible from the entry. How can it do? It binds to the ACE2 receptor on the cell membrane, so the entry of the virus can occur. So we also know that SARS-CoV-2 spike protein is heavily glycosylated. The infectivity can be inhibited by lectins, and this done by specifically binding to the glycans of some receptors, we can say. And another point, this about the lectins is the mannose binding lectins interfere with coronavirus entry by binding to high mannose type of N glycans of SARS-CoV with the S protein. So the main focusing protein is, we can say, S protein. And by binding or targeting this protein, you can prevent the viral attachment to the host cell. So this is a maybe short table of giving information, the structure classes of the lectins, and some glycan specificities of these lectins, and the glycoprotein that they bond. So these both lectins in this table have some different antiviral activities, and this is studied up to now. So let's give some examples of the specific lectins that are studied with this new SARS-CoV-2. One of them is grifixin. This is an important antiviral lectin. Maybe it can be said that the most potential antiviral lectin up to now. It is isolated from red algae, and I put this manuscript. It is maybe published published online 2 or 3 days ago, and they found that this potential lectin is also inhibits by targeting the SARS-CoV-2 spike protein sub-2 subunit. So this is the most important lectin, we can say, up to now. Also, another study, another carbohydrate binding protein from a different beans, edible lab beans. The lectin name is FRIL. This is a specific lectin. It binds to interacts with FL3 receptor on the membrane, but it is found that it has a potential activity, antiviral activity against SARS-CoV-2 and also influenza virus strains. So this is also in the literature a valuable lectin, we can say. It is also mediates the action by interacting with the complex type N glycans of the viral glycoproteins. And also another examples, and it is a new article again. It is also a plant lectin, concavalin A, and again it targets with the N-linked glycans in the spike proteins, and it causes antiviral activity, we can say. So thank you very much for your attention.
Is there any question or any comment? No?
No question, but just a comment for maybe. May I?
Evet, Nazlı Hocam dinliyoruz.
Yes. We found that, or we know that, especially from today's and the yesterday's presentations, licorice has a potential use of treatment or blockage of the SARS-CoV virus. So maybe it's important to find out its lectins. This is first point. And the second is lectins are sometimes harmful for humans because they are carrying some parts or some specific action on enzymes. So we need to check their toxicity and we need to check their beneficial effects on human. Thank you.
Okay. I can also maybe answer these comments about these comments. Also, I read a literature about this antiviral activity possessing lectins. They treated the rats, infected rats, and they also see some important. They saw that they lose a lot of body weight. This is the disadvantage, maybe. You are right. Also, clinical studies should be done because these lectins have a lot of toxic also properties, or yes, clinical studies should be performed.
Is there any question? Any more? I think no question. I would like to thank you. I'm closing this session. I would like to thank you, chairs of the session, for Professor Violina from Bulgaria and Narin from Turkey. And we will start the next session afternoon, 1 p.m. Istanbul time. Please keep signed before 5 minutes. And I'd like to announce here, we publish MESMAP-6 poster presentation on our YouTube channel. You can enter our MESMAP-6 YouTube channel and follow the presentation and stop and play. You can easily check. And if you have any question, you can ask the authors in the poster session. Thank you again for your kind participation.
Thank you. Thank you,
Thank you.
Nazım, for great efforts for us. Thank you.
Thank you very much.